So let's say you eat a slice of bread. How do you turn that bread into energy? Well, it begins with these things called macromolecules, which are just large organic molecules. Bread is mainly made up of carbohydrate molecules, which are sugars and starches. Glucose is a simple sugar that is a component of many carbohydrates. The glucose molecule is an important source of energy for organisms. In order to get energy, you have to break down glucose and convert it into a molecule called adenosine triphosphate, or ATP. ATP is a primary energy storing and transferring molecule and is often called the currency of biological energy. A lot of energy is stored in the bond between the second and third phosphate group. In order to get ATP from glucose, your cells undergo a process called cellular respiration. The first part of cellular respiration is a process called glycolysis. Glycolysis is an anaerobic process, meaning that it does not require oxygen in order to work. This process takes place in the cytoplasm of the cell. In glycolysis, two ATP molecules are actually invested in order to give glucose two phosphate groups. This is called phosphorylation. Because the ATP molecules each lose a phosphate group, they turn into ADP, adenosine diphosphate, which contains less energy. That's okay though, because it can later be converted back into ATP for more energy. Glucose is shaped by enzymes and redox reactions, which is short for reduction-oxidation reactions. In redox reactions, a molecule loses an atom or group of atoms, and another molecule gains those atoms. The oxidized molecule loses, while the reduced molecule gains. You can remember this with the acronym OIL-RIG. Oxidation is loss, reduction is gain. In glycolysis, NAD+ is reduced, forming NADH and releasing energy. NADH will be useful later on in cellular respiration. As I said earlier, the two ADP molecules are converted back into ATP. This is done through a redox reaction. Two additional ADP molecules are converted in glycolysis, each taking a phosphate group phosphoenolpyruvate, glucose that underwent redox reactions, to produce two ATP molecules and two pyruvate molecules that each have 3 carbons. There are many other reactions that take place in glycolysis, but I won't go into detail on all of them in this video. Having many reactions that release energy in small amounts means that we can capture more energy than if we released all of the energy at one time. At the end of glycolysis, there is a net production of 2 ATP, 2 NADH, and 2 pyruvate molecules. These products will later be used to make even more ATP. Next comes an irreversible process called pyruvate oxidation. Both of the pyruvate molecules produced in glycolysis undergo this process. It is important to note that pyruvate oxidation takes place in the cytoplasm and in the matrix of the mitochondrion. Pyruvate reacts with oxygen to produce a 2-carbon molecule called acetate and CO2. NAD+ is then reduced by coenzyme A to make NADH. The The oxidized acetate molecule then bonds with coenzyme A to form acetyl-CoA. Acetyl-CoA is needed for the next process of cellular respiration. The end products of pyruvate oxidation are acetyl-CoA, NADH, CO2, and H+. The third process of cellular respiration is the Krebs cycle, also known as the citric acid cycle, and occurs in the matrix of the mitochondrion. Unlike previous steps, the Krebs cycle requires oxygen. The cycle begins when acetyl-CoA and a molecule called oxaloacetate combine to form citrate or citric acid. Citrate is then isomerized, which is a fancy word for rearranging the atoms of a molecule to form a slightly different molecule. Like we saw in previous steps, NAD+ is reduced to NADH. This happens numerous times in the Krebs cycle. A series of redox reactions take place, changing molecules slightly each time until we have oxaloacetate and can begin the cycle again. NADH, FADH2, and ATP are produced during these reactions. The final process of cellular respiration is the electron transport chain. Protein complexes are embedded in the membrane of the matrix in the mitochondrion. NADH gives a proton and electrons to the first protein complex, NADH dehydrogenase. NADH dehydrogenase pumps one proton per electron through the membrane of the matrix, using some of the energy from NADH's electrons. The rest of the electrons are passed on to ubiquinone and are transported to the cytochrome BC complex. Ubiquinone only takes enough energy to be able to transport back and forth between the two complexes. Cytochrome BC pumps more protons out of the matrix and passes the electrons to cytochrome C. Like ubiquinone, cytochrome C only takes enough energy to transport between complexes. It passes the electrons off to cytochrome oxidase. Cytochrome oxidase pumps protons through the membrane of the matrix, but also reacts with oxygen to form H2O and release it into the matrix. This all creates what is called a proton gradient, which just means that there is a higher concentration of protons outside of the matrix than inside of the matrix. The protons re-enter the matrix through ATP synthase, as they naturally want to go back into the matrix in order to even out the concentration. The protons contain potential energy, which is used to form a bond between ADP and PI, which is inorganic phosphate. The electron transport chain produces a total of 32 ATP molecules. In total, cellular respiration generates up to 36 ATP molecules per 1 molecule of glucose. Thanks for watching!